Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the series on food microbiology. In today's lecture, I am going to talk about the extrinsic parameters which control microbial growth. In the last two lectures, we saw how the pH, the oxidation reduction potential, the presence of antimicrobial components, so many other factors which influence the microbial growth in the food, and what can be utilized to help to preserve the food component concerned, limiting microba. Here I am going to talk about the external factors, the extrinsic factors, which these are the properties where of the storage area, the storage environment, which affect not only the food but also the microorganisms which grow in them. The temperature of storage, the relative humidity of the environment, the presence and the concentration of gases, the presence and activities of other microorganisms. Temperature. As we have been seeing from the beginning, each microbe or each organism has a specific temperature where it will have optimum growth. This will not be a specific temperature like 27 specific or 28 specific, but normally this will be a range. So, when we consider that range, the lowest possible temperature where microbial growth has been reported is minus 34, and the highest is somewhere in the excess of 100 degrees centigrade, which means that these microbes are capable of surviving in temperature as low as minus 34 or in temperatures as high as 100 degrees centigrade. Microbes have also been reported and found out, people have discovered microbes even in the magma, in the volcanic vents, which means that organisms capable of surviving that high temperatures are also present. So, but here when we talk about spoilage microorganisms and storage microorganisms, we need to consider for each particular type of food, what is the type of microbe which causes maximum spoilage or damage, what is the ambient temperature in which it can grow very well, and then preserve the food in a different temperature which will prevent the microbe from growing or proliferating. So, that is how we categorize these. So, for example, we call one category of organisms are called as psychotrophs. These have actually a optimum growth range between 20 degrees centigrade and 30 degrees centigrade, okay? But they can also grow below at or even 7 degrees centigrade. So, these we call them as psychotrophs. Those that grow well between 20 and 35, that is maximum growth would be between 20 degrees centigrade and 45 degrees centigrade. The optimum growth would be between 30 and 40, but by 10 degrees centigrade less than 30, that is at 20 degrees, and 5 degrees above or at 45 also they can grow, but their optimum growth, their maximum activity would be between these 30 degrees and 40 degrees. These category of microbes, we call them as mesophiles. Those organisms which grow well above 45 degrees centigrade, we call them as thermophiles, but for these thermophiles, most of them well known thermophiles, they have an optimum range of growth between 55 and 65. So, psychotrophs have an optimum between 20 degrees centigrade and 30 degrees centigrade. The mesophiles have an optimum between 30 degrees centigrade and 40 degrees centigrade, and the thermophiles have an optimum between 55 degrees centigrade and 65 degrees centigrade. Now, most of the microbial growth is accomplished by enzymatic reactions, and as you all know, enzymes have their maximum activity at specific temperatures. So, within a certain range, with every 10 degree rise in the temperature, the catalytic rate of an enzyme will double. So, an enzyme can have a sluggish activity at the less than optimum temperature, maximum activity at the maximum optimum temperature, and then it will slowly, as the temperature increases, the enzyme will slowly get denatured. So here we are talking about the microbes whose enzyme activity actually will decide how fast the food is going to get degraded, because the organism can grow in the best possible aspect because it is at the optimum temperature. At optimum temperature, the enzymes are very active, so the food will get spoiled very fast. Less than optimum, the microbes still survive, the enzymes do act but at a slower rate, and as the temperature increases, the activity will slowly reduce to the level that ultimately the enzyme gets degraded when the temperature goes beyond a particular level. This enzymatic reaction is reduced by half by decreasing the temperature by 10 degrees centigrade, and this relationship changes beyond the growth range. The influence of temperature on enzymatic reactions play an important role in microbial growth in food. So, when we are going to store the food, first we understand that what are the types of microbes which can cause spoil to that particular food component. 2, the optimum temperature range in which the microbes grow, that is the enzymes of the microbes which show the maximum metabolic activity to spoil the food concerned. So, if these two facts are known, then your storage temperature now can be either below or above these two optimum temperature in such a way that the spoilage organism does not proliferate in the particular food concerned. The food gets preserved for a longer time. Food is exposed to different temperature from the time of production till consumption, meaning depending upon the time during its growth. For example, a plant from seed germination to where it becomes a vegetative plant to starting it sets the seed, and then you process it, or you are going to get the vegetable or the fruit. So, for each it would have been exposed to different temperature. Then when it is after harvest, when it is going to get processed, then also you will have a variation in the temperature. So, depending upon the processing condition, the food can be exposed to high heat, meaning 65 degrees centigrade. For example, roasting of the meat is done at a very high temperature, to more than 100 degrees centigrade, where we talk about preservation of the food in ultra high temperature processing. So, this change, exposure to low heat, to medium heat, to high heat, to very high degrees exposure, this can kill or inhibit a lot of microbes which are naturally present in the food. So, when those microbes get killed, automatically we will have long term preservation of the particular food concerned. For long term storage, the food can be kept at 5 degrees or to minus 20 degrees centigrade or to below freezing. In all these conditions, the microbes will have either less activity or absolutely no activity, as a result of which the food will get preserved. Some relatively stable food are also kept between 10 degrees and 35 degrees, which we call as the cold to ambient temperature. And some ready to eat foods are kept at warm temperature, meaning 50 to 60 degrees centigrade for several hours, for example, in the supermarket deli. Different temperatures are also used to stimulate desirable microbial growth in food fermentation. Now, here we have two aspects. One, control of the microbes which spoil the food. Two, allow the microbes which cause fermentation, or because of microbial growth you will have certain flavors which will be produced in the food which are acceptable to the consumers. So, if you want to create that flavor, then the food has to be kept at the optimum temperature or the ambient storage condition, when those organisms can grow, produce the enzymes or the metabolic reactions which produces that flavor. So, this again depends upon the type of food we are trying to talk about, and also the nature of the flavor which is accepted by the consumer. Microorganisms which are important in foods are divided into 3 groups based on the basis of temperature of the growth, each group having an optimum temperature and optimum range of growth. As we saw earlier, the thermophiles, which grow relatively at high temperature with optimum between 55 degrees centigrade and 45 degrees centigrade to 70 degrees centigrade. The mesophiles, which grow at ambient temperature with optimum at 35 degrees centigrade and a range of 10 to 45 degrees. Then the psychrophiles, which grow at cold temperature with optimum of 15 degrees and a range of -5 to 20. So, in all these cases the optimum is one value, but the range is a broader area. So, depending upon this broad range is where we consider food preserving or storing temperature. When the food are exposed to temperatures beyond the maximum and the minimum temperatures of growth, the microbial cells will die at a higher temperature and relatively slowly at a lower temperature. So, this growth in and viability change is important in reducing food spoilage and enhancing safety against pathogens as well as foreign food processing. So, here as you saw, you have a range of temperature from 10 to 45, or what I talk about 45 to 55 or 65. So, the organism is viable and growing at that temperature, maybe not in the optimum activity, but it will still be present and it can still cause damage, maybe at a lower rate. But as the temperatures go beyond this, the organism will die. So, this in turn will help to kill those organisms, thereby increasing the shelf life. That is one aspect, thereby you are reducing the foodborne pathogens or you are reducing the amount of spoilage organisms in the food. The second aspect, as I talked about, is food bioprocessing, where you want to produce certain nutritive components in the food, enrich the food because of certain bacterial flavors or enhancers which are naturally synthesized in the microbe. Then you require those microbes which you produce these to proliferate at a higher rate. So, the range of temperature where you store or to preserve the food should be optimum for the growth of those. Then this would become ideal. Relative humidity of the environment. Relative humidity and water activity are two things which are closely related. When a food has a low water activity, for example, you dehydrate, the seed is dehydrated, grain is dehydrated. Okay? So, then at low the water activity, the microbes cannot grow. But if the relative humidity where the food is stored or the grain is stored is high, then the moisture present in the atmosphere in turn can be accepted by the dehydrated food, thereby increasing the water activity. So, over a period of time, what will happen is that the bulk of the grain or the bulk of the food with the low water activity will absorb the moisture from the humid environment and slowly show increase in the water activity. But within that bulk, there would be isolated pockets where you will have high water activity at a higher rate, and these will create pockets, and higher water activity automatically triggers microbial growth. So, what happens is that as the moisture gets absorbed by the food, slowly depending upon the level of moisture which gets absorbed, you will have pockets of microbes which get activated. So, that particular area, that particular square feet of the food will show higher microbial growth, higher activity. So, slowly over a period of time this will spread throughout the food concerned. So, even if to begin with the food had a low water activity, because of the relative humidity present in the environment, slowly the water activity will increase, thereby increasing the microbial attack and spoilage of the food concerned. The next aspect we talk about are halotolerant, osmotolerant and zero tolerant organisms. See, normally in food preservation we can use high amount of salt, high amount of unionized organic compounds like sugars and completely desiccate or to produce dry food. In all the three conditions normally microbes cannot grow, but you do have organisms which can still withstand this high concentrations. For example, those microbes which can grow in the presence of high concentrations of salt, we call them as halotolerant. Those which can grow in the presence of sugars, we call them as osmotolerant microbes. And those which can still grow where your water activity is minimum in the dry food, we call them as zero tolerant. So, microbes are capable of adapting to any sort of environment because evolution has given them that advantage. This table actually shows the water activity of various types of food. You have the dehydrated food with extreme low water activity, with the dried fruits, caramelized components giving the next, with pickles and jams coming to the level, then you have the cheese, flour, etc., which have a higher level of water activity. Then comes your meats and etc., followed by fresh vegetables, fruits, milk, etc., which are high in water activity. So, from a grade, these are the ones which will get spoiled fast. In a decreasing scale, the spoilage would be like this. When we store food, as we saw earlier, you have aerobes and anaerobes, those which essentially require oxygen in the environment for their growth, those which do not require, where oxygen is inhibitory. Then you have the in between ones, facultative aerobes or facultative anaerobes. And you do have organisms which also depend upon the other type of gases, the gaseous mixture which is present in the environment. For example, carbon dioxide. The inhibitory effect of carbon dioxide on microbial growth is applied to the modified atmospheric packaging of food. You have a sort of packaging, a different type of packaging which is called modified atmospheric packaging, where oxygen concentration is reduced, the food gets packed in an atmosphere which is high in carbon dioxide. So, under this condition, because of the higher amount of carbon dioxide which is present during the packaging condition, the microbes will get inhibited. The second gas which is antimicrobial is ozone. This is also used to increase the shelf life, but the ozone has an issue because this is a very strong oxidizing agent. So, if the food contains fatty acids and or high amount of storage lipids, then ozone can cause rancidity. Otherwise, it is a very strong antimicrobial and it can destroy almost all the microbes which are present in the food, thereby increasing the shelf life of the food concerned. The next category which will inhibit food spoilage is the presence and activity of other microbes. For example, some microbes which are already present in the food will inhibit the growth of other microbes. For some foodborne organisms produce substances which are lethal or inhibitory to others. These components which are secreted by the microbes, we call them as either antibiotics or bacteriocins. For example, some will produce hydrogen peroxide, some produce organic acids. But whether it is an antibiotic or a bacteriocin or a hydrogen peroxide or an organic acid, each one of them have an inhibitory capacity to go and inhibit the other microbes which also dwell in the same food. So, essentially the microbe which produce this is creating an environment where it alone can utilize the nutrition present in the particular food concerned, and it is eliminating its competition by being lethal to the other microbes which can also essentially use up the same nutrients. There are two categories here. One is called as the general microbial interference. Here it means general non-specific inhibition or discretion of a microbe by the other. Here what we are trying to explain is the growth of one microbe not because of a specific pathway or a compound, but because it is growing by a general non-specific inhibition, it will automatically go and reduce the other microbes which can also grow in the particular food concerned. An example to this is the suppressive effect of a large number of aerobes against the growth of Clostridium botulinum in fresh meat. Clostridium botulinum, as you all know, is a gram-positive spore-forming organism. The spores of the Botulus toxin is extremely lethal. It can cause instantaneous death. And as you know, bacterial spores are very difficult to kill. You require extreme high temperature and pressure to gather. For example, autoclave, that 121 degree Kelvin, starting at a high pressure for 20 minutes is required to kill the spores, as you have all know. So, and Clostridium botulinum is normally present in meat. So, if that has to be reduced, if the meat sample has other aerobes which are growing, a large amount of such aerobes in turn can inhibit the slower growing Clostridium botulinum present in the meat. The second aspect is what we call as the lactic antagonism. Here we are going to talk about the bacteriocins, the pH depression, the organic acids, diacetyl. All of these components can inhibit pathogen and the food spoilage organisms. Let us see how this works. First is the general interference. Here I said that the general interference will work if you have a large number of the background microbiomes which in turn can go and inhibit the lesser number of the spoilage or the pathogenic organism. So, that ratio has to be maintained, a large number of microbes targeting a smaller amount of either spoilage organism or the pathogenic organism. And secondly, the interfering microbiota. They should not be a homogeneous in composition, it should be a heterogeneous composition containing various genera and mixed species. So, such a composition with the background microbiota in large number, with the spoilage or the pathogenic organism in lesser number, that comes under what we call as general interference. And this in turn can be either by competition for nutrition, because if the organism, the pathogen or the spoilage organism and the background microbiota utilize the same carbon source, the same energy source, then because of the large number of the background microbiota, the smaller number of these cells will not be able to get the sufficient amount of nutrition, which in turn will reduce their growth rate. The second is they have the same competing for the attachment or adhesion site. To attach or stick onto a cell, they have the receptors. So, if these two organisms compete for the same attachment site, because of the sheer number, the background microbiota will win against attachment site. Then unfavorable environment, which can be any of the conditions, or it can be all the three together. So, when all the three factors come together, the background microbiota will win, thereby inhibiting the spoilage microorganism or the pathogenic organism. So, this is the pattern in the general interference caused for the microbial destruction. Nowadays, instead of taking the extrinsic factors alone or the intrinsic parameters alone, we consider both the factors together. That is, we take the nature of the food product concerned, take the conditions, take the microbiota. One, the good microbes which are normally present in the food. Two, the spoilage organisms. Three, the pathogens. Then, the ideal conditions for the growth of all this. So, we are going to combine both the intrinsic factors which can promote microbial growth, the extrinsic factors. Then we take the optimum amount which will cause the good microbes, which will cause the bad microbes to die fast, and combine both these factors. This is actually called as the hurdle technology, where multiple parameters are combined to control the microbial growth in food and also to increase the shelf life. In nowadays, in food biotechnology as such or in food microbiology as such, when we talk about packaging, increasing shelf life, or we talk about extending the nutrient component of the food, we talk about this hurdle technology, where we combine the intrinsic and the extrinsic parameters, look all the factors together, and then taking these two together, we will control the microbial growth. In this session, we saw how the intrinsic and the extrinsic parameters can be used together, how the good bacteria, those which promote the good aspects of the food, as well as how to control the spoilage microorganisms or the pathogenic organisms, how to preserve or increase the shelf life of a particular type of food by promoting the natural antimicrobial aspect of the particular food concerned, and how to combine using a hurdle technology the preservation of food particle concerned. Thank you.